Thank you for the invitation to speak today, and also for the introduction. As was just mentioned, the site where I work is concerned with all aspects of projects, ranging from early phase development through to on-market technical support and manufacturing of established products. And what I'd like to do in my presentation today is give three real-life examples of where we've had to carry out impurity or contamination investigations, and particularly how we've used NMR in these investigations. Before I get on to that, I think it's useful to just do a quick recap of NMR as an analytical technique, certainly in this perspective. And the main thing to point out about it is its inherent lack of sensitivity compared to other techniques that we might be familiar with. This is by virtue of the fact that it's a very low energy technique. We only ever get a very small population excess between our ground state and our excited state nuclei to generate signal. And what that effectively means is we only ever really see a very small percentage of the sample that we want to analyze. On the plus side though, it's non-destructive and it allows us to see samples and observe samples in a non-perturbed state. It's inherently quantitative as long as we follow certain protocols. That's very powerful for getting accurate measurements on things. And it can be very selective for a range of nuclei and physical forms, and I shall certainly be exemplifying that in the examples that follow, allowing us to look at only solution species and specific nuclei within our sample. So applying that thinking to the first example, and this is not an instance where we had contamination of a product or a medicine, but actually contamination of heated chiller oil in a freeze dryer that we use for manufacturing one of our established products. So we've got no issue with quality, but we have got a potential impact on supply to patients here because the instrument or equipment is not performing as it should. The engineers hypothesized that they were getting leaking of the refrigerant of the freeze dryer into its circulating heated chiller oil, and they wanted to know if NMR would be able to help with testing that hypothesis and detecting what they needed to detect. As you can see on this slide, it's an example that lends itself really nicely to solution state NMR analysis. We were given really large samples of material, so we're not sample limited in any way, which is fantastic. And they are liquid samples, which lends itself perfectly to the physics of NMR as a technique. So we were able to analyze neat samples without the need for any kind of dissolution or preparation in any way. And you can see here that the refrigerant that we're probing for in this case, R404, is a mixture of different fluorinated hydrocarbons, and we're wanting to see if they are potentially present in mineral oil. And this gives us fantastic selectivity to fluorine NMR here to selectively probe for these contaminants, and the rest of the sample becomes effectively visible to us for it. And on this slide, you can see the outcome of the investigation, really quite straightforward to carry out, very simple to report. At the bottom here, you can see a reference fluorine spectrum for a reference of mineral oil taken straight from drum supply. And you can see that it is effectively a fluorine species free. Similarly, an example of mineral oil that was taken from a well-functioning freeze dryer that had no issues, you can see has the same profile. But comparing that to the mineral oil drawn from the suspect freeze dryer, you can see that it's significantly contaminated with a number of fluorinated species. And that was all the information that the engineers needed in this instance. We didn't have to carry out any kind of quantitative analysis. We didn't even have to identify what the species were. We've proven the hypothesis of the leak of those materials, and that allowed them to effect a repair and get the patient supply back on track with no risk or no consequence. Moving on to, excuse me, my second example. This is probably something that people are a little bit more familiar with when it comes to purity contaminant investigations. And this is the synthetic step of a drug compound intermediate that's carried out as a CRO for us. And both within the synthetic process and also in the isolated product, they've detected higher than normal levels of a high molecular weight species. And you can see we are talking trace amounts here, but I think really underlines the importance of the levels we have to go to. Some early investigation work concluded that the impurity that they were seeing in this intermediate was actually related to a related impurity that was present in the starting material. Very quickly became the focus of investigation. Some initial UPLC-MS analysis gave a molecular formula, a molecular weight, and from that we were able to propose a structure, a tentative structure for this impurity. Came up with this phosphorus-linked pseudo-dimeric structure, so something closely related to the desired material, is phosphorus linking. Now, at the time that we started the investigation, the only sample that we had available was the bulk starting material with this unknown impurity present at about 0.1% level. And we were asked if that would be okay to initiate an NMR investigation. And normally we would say no to that. We want to elucidate or confirm a structure using NMR. Normally ask for material to be provided in at least 80% purity so that we can have confidence in the signals and the correlations that we're observing in the data. However, in this case, we were happy to make at least a start because again, got the selectivity of being able to probe specifically for the phosphorus nucleus within the sample, effectively make the bulk material invisible. So with that in mind, we carried about a couple of experiments that you might expect to do. Again, we're not sample limited, which is great. We have great solubility, so we were able to make up a really nice strong sample of our material, which helps us overcome some of the sensitivity issues. And first experiment, as you might expect, 1D phosphorus NMR identifies that there are in fact 2 phosphorus containing species present in our bulk material, one of them in a more significant level. And additional to that, we carried out a proton phosphorus HMBC experiment, and that allows us to link this phosphorus content to signals that we can potentially now go and hunt for in our proton data. At this point, we're also able to make a modification to our proposed structure, and this is the kind of thing that always evolves in these kind of situations. If I just go back a slide to our original proposed structure, you can see we would have expected to see one correlation in our HMBC data from this CH position here correlating to the phosphorus center. And the fact that tells us we need to rethink that, and we very quickly came up with this alternative proposition. It's not linked through a CH, then maybe it's linked bridging these nitrogens. That was a much better fit to the observable data and also some predicted data structure. Now that we're armed with this information, we can go digging in the proton data with more confidence than we might be able to in normal circumstances. Like I said, if you're looking for trace amounts of something in a parent compound, you can't really have confidence if small signals like this relate to the impurity that you're interested in, or a different impurity, or just something else that happens to be present in the system. But with that phosphorus selective 2D data, we can now say with great confidence that this signal here and this signal here relate to our impurity. And because we've got really nicely resolved signal here, we can integrate that accurately against the carbon-13 satellite of the parent material and get a really nice accurate measure for the level that's present in the parent compound, and it was determined at 0.1 mol%. So that's a really useful piece of information to have. Finally, the last thing to point out on this slide, with this new proposed structure, we would expect to see a third signal. We'd expect to see a signal CH here between these 2 nitrogens. And we've got what we think is a likely suspect here, but again, we're scuppered by the fact that it's present in such low levels. We have no correlation to 2D data, so we can't be confident in this piece of data just as it stands. And we move on to what is always the ultimate goal with investigations like this, and that is isolation or synthesis of a pure sample of the representative material. And the chemist did a really good job with this, provided me with a solid sample, and I was able to dissolve that in DMSO, and then, as you can see here, go through the usual process that we go to establish the structure of a compound, gathering the usual 1D and 2D NMR data, tying it all up nicely together in Spectra's processor to give you a single object that allows you to confirm and verify that structure. You can see I'm showing the HMBC data here as an example. And that's really the last piece of the puzzle. we've confirmed the structure, we can relate these signals back to the signals that we're observing in our impure sample. We can also carry out spiking experiments now and really be absolutely confident that that is the structure of our impurity. With one final little twist, and that was the question around, were we observing the POH compound as had been determined by UPLC, or were we actually observing the trichloro compound? There were two ways to approach this. I was happy fairly quickly actually to determine that it was a trichloro compound. I was given a nice dry sample and I used anhydrous solvent to do the analysis. You can see here that there is only a small signal annotated that corresponds to the OH, the POH compound. And I was happy to confirm that the bulk material was trichloro, now that we were observing it in a non-perturbed state. Mass spec also went back and had another look at this using a new ionization technique where they sampled directly from solid rather than dissolving and subjecting the compound to hydrolysis. And again, here you can see the detected isotope pattern and also the predicted isotope pattern for the trichloro compound. Complicated slightly in this case because we have a mixture of M plus H and M plus H plus, sorry, M plus and M plus H plus mass ions, but however it holds true. And then once we have that structure, we obviously understand its formation, we understand the consequences of it being present in our material. I can make sure that we control and manage any risk associated with that. The last example that I want to talk about is something a little bit newer, something a little bit earlier, and it relates to a new product that we're currently developing, ACDLabs, and this is a drug dendrimer conjugate product that consists of a fifth generation pegylated lysine dendrimer, which has got a number of small molecule warheads attached to it covalently via a hydrolyzable. Now, the final synthetic step involves amide bond formation to attach this warhead, and that happens in the presence of free TFA that's in the system as a result of a previous deprotection. And so there was the hypothesis, there was the belief that we were potentially getting TFA covalent bound to this construct in a site or sites, and that could then potentially hydrolyze when the drug is formulated for administration, and could potentially lead to risk of exposure of TFA to patients. So regulators asked us to get a better handle on this, understand identifying that species and understanding its kinetics and how to control it. And I was asked if I would be able to do this using a reaction and process monitoring technique with NMR, which is something that I spend a considerable amount of my time working on. And what they wanted to know was, could they observe loss of this TFA or this hypothesized TFA added signal to growth in free TFA to serve as both proof of structure and also to understand something about the kinetics. And then you can see in aqueous media, so this is the fluorine method that we use for release of drug batches. It's in DMSO, and in organic media, O466 behaves pretty well like a small molecule. We've got signals here for the internal standard, slightly broadened, still discrete and measurable signal for O466, which comes from on the warhead. Free TFA, which we're able to be confident in using spiking, and then this other signal for believe is TFA adduct. And can we do kinetics and link those two together? We want to understand this behavior though in the aqueous medium that's used for its formulation and its delivery. And here we come across a problem that we've always had trying to analyze O466 using NMR, and that is that it's very insoluble, suffers from a lot of aggregation effects. And so the quality NMR data that you see in organic media really go out the window, and we struggle greatly we can still speciate things, but we lose signals. You can see we've now can no longer observe the signal for this proposed adduct, and we've lost resolution between signals for the construct itself and the free TFA. However, we can still see and measure the free TFA, and so we undertook experiments to see if we can just track the change in behavior of this signal and see if that tells us anything about the system. So we made up samples of 0466 in its clinical administration medium, and that's an aqueous pH 5 buffer. It was then put into the NMR spectrometer at 30 degrees. That's the upper safety limit for preparation of samples. And then over increasing time periods, you can see we ended up having to go quite long to fully observe and fully understand the system, measuring and integrating and plotting the change in the free TFA signal. You can see really quite nice classical predictable observable behavior for the release of TFA. And we were able to plot this, as you can see, and then because we're getting inherently quantitative data, we're able to compare this to the signal for a concentration, a sample of no concentration, and relate this data, plot it as a function of ppm of 0466, which is a specific measurement that regulators asked us to make. And once we were armed with this information, we were then able to do two really powerful things with it. Firstly, we were able to further validate the fluorine assay method we were already using. You can see on this plot that you're able to read off quite easily a level of free TFA that is present at T0, and we found that that agrees really nicely with the level of free TFA that can be measured from integrating this signal here. And you can also see that the data trends towards an ultimate concentration or total TFA content once everything is hydrolyzed, and that tied in really nicely with the measure of TFA that could be inferred by measuring this signal here. So we were able to identify this signal as the hypothesized adduct, and also understand from that how much adduct was present in any batch of drug product. Ultimately, what we were able to do with this was one of my kinetics colleagues was able to use this data to create, validate a model to predict hydrolysis of TFA adduct to release TFA under a variety of different biological conditions. And that means that we can now measure or assay any batch of 0466 using our established method and understand from integration of this signal here and application of this model ultimately what the risk of exposure of TFA to a patient will be. And then again, that allows us to control that and remove that risk, so there is no danger to. And that's it. That's the end of the three examples. Hopefully, I've shown how NMR has been particularly useful in determining the structures, determining the root cause in all of these cases. Really critical to point out that we've managed to control patient safety in every case here, either preventing material from being administered or being exposed, so prevent a risk. And in the process of achieving our primary goals, we've also learned additional things about some of the systems, which is useful information as well. As other people have commented, these things are always collaborative efforts between chemistry, analytical, and the different specialists, and these examples were no different, even though I've only spoken really specifically about my involvement in them. So I have to acknowledge the other specialists and chemists and analysts that contributed to the examples I've shown. And that's everything I had to say.
Okay. All right. Thank you so much. So we can go ahead and take some questions now. Mm-hmm. So with respect to the freeze dryer example, so using fluorine NMR there had a clear selectivity advantage, but what were the implications in terms of sensitivity?
Okay, yeah, that's a really good question. Let me go back to my introduction slide. So if we think about the concept of receptivity with NMR, this is a combination of the energetics of any given nucleoside, the difference in these energy levels, and how good a population excess you can get, and also considers the natural abundance. And you can see the best case is for proton, and so we calibrate scale receptivity to 100 for proton. Now, all of the nuclei suffer relative to that, but we can see that in the case of fluorine, it's actually it's not that bad. It's 83% as good as proton, basically. So we are taking a slight hit on sensitivity, but not by much. And the other thing that manufacturers consider this, and so the probe, the instrument that I use to do that particular piece of work is manufactured to enhance fluorine sensitivity above anything else. So it works in our favour here, and then also manufacturers also boost the sensitivity when they can. Fluorine is particularly powerful. Not only is it a good nucleus for NMR, when you consider that it's also present in 25–30% of current small molecule drugs, it means it's a really nice, effective and selective handle for a lot of the compounds that we're studying in a minute.
Okay. As in a similar vein, I think all of the examples that were shown today relied on heteroatom nuclei for detection of these impurities. And so is that pretty typical that you're relying on a heteroatom for that selectivity?
I would say no, actually. Again, because we know we're always going to take a hit compared to proton. We are always striving for receiving that material isolated or synthesized to a high purity, so that we can look at something neat. And we don't need much. If we have a few mgs of material, we're able to do all of the usual 1D and 2D NMR data that we need. But we are still using the traditional experiments to do structure elucidation, proton NMR, carbon NMR, 1D and 2D, because they are the most abundant nuclei, and that's what you need to establish that connectivity and establish that structure elucidation. The heteronuclei are useful from a selectivity point of view, but they're so sparse, or can be so sparse in the structure, that they don't give you the complete picture.
Okay, I guess that leads pretty well into the next question as well. In the API example, can you comment on like how important it was that you elucidate that full structure of the impurity?
Yeah, and I've been helped out here by Andrew's talk previously, and he highlighted a couple of the important things. If you can identify the structure unambiguously, then it means you can have a really good understanding of how it's arisen, where it's come from, and that allows you to put control in place to either limit its formation or its formation. So that's a really powerful part of it. And then the other aspect of it, and I know we have a presentation later on coming up about assessment of mutagenicity or potential mutagenicity, sorry. Once you have that structure, you're able to carry out those theoretical screens and get a handle straight away whether or not this is something that is a potential mutagen. Do we have to control it to even more stringently?